Today's presentation is Accelerating Vaccine Development, Applications to COVID-19 and Future Pandemics. Next slide. We would like to thank the American Society for Clinical Pharmacology and Therapeutics, as well as Pharmacokinetics Africa, for their generous support of this webinar. Next slide. Today's speakers are Drs. Thomas Kerbusch, Amy Chung, Joost Lemers, and Pete Van de Graaf. Thomas is currently Certara's Chief Growth Officer. In this role, Thomas drives integrated growth opportunities with a special focus on key account management and client relationships. He has more than 20 years of experience contributing to the development and approval of numerous drugs in a range of therapeutic areas while at Pfizer, Organon Schering-Plough, Merck MSD, and Certara. He holds a doctorate in clinical pharmacology from the Netherlands Cancer Institute and has co-authored over 40 peer-reviewed publications. Next slide. Joining Thomas is Amy, who's currently a senior director at Certara's Integrated Drug Development Group. She has over a decade of experience in clinical development in the pharmaceutical industry. She earned her doctorate and also completed a postdoctoral fellowship at the University of Manchester on structural identifiability and mechanistic modeling. Prior to coming to Certara, Amy was previously a senior clinical pharmacometrician and project manager in the pediatric working group at AstraZeneca. She's also chair of the IQ Consortium on Clinical Pharmacology and Translational Medicine and ADME Sciences Leadership Group, as well as the PBPK Pediatric Working Group. Next slide. Joost Lemers is also currently a director at Certara's Integrated Drug Development Group. He has more than 20 years of experience in the pharmaceutical industry using computational methods, statistics, and simulations to support decision-making. He has a doctorate in bioorganic chemistry from the University of Utrecht in the Netherlands. Prior to coming to Certara, Joost previously worked in the Department of Clinical PK/PD at Merck MSD. He's an expert in supporting pediatric, vaccine, and metabolic disease development programs. Next slide. Lastly, we have Pete Van de Graaf, who's currently a Senior Vice President of Quantitative Systems Pharmacology at Certara. Pete has more than 20 years of experience working in the pharmaceutical industry at Sanofi and Pfizer. He holds a PharmD from the University of Groningen and a doctorate in clinical medicine from King's College London. Pete is a fellow of the British Pharmaceutical Society and also is concurrently a professor of systems pharmacology at Leiden University in the Netherlands. Pete is also the current editor-in-chief of Clinical Pharmacology and Therapeutics. Next slide, please. Thomas, Amy, Joost, and Pete, welcome to today's event. I'll now turn it over to Thomas to begin the presentation.
Thank you, Suzanne. Let me just take control here. Let me just give an introduction on what we're going to talk about for today. Many of you have seen this graphical illustration, and beyond a shadow of a doubt, vaccines are among the most effective medical interventions that we have in saving human lives and avoiding suffering. And it doesn't mean that we always get it right, though. And so here's an example of the seasonal flu vaccine, where over the years, the track record has been stronger and less strong as well in preventing flu, hospitalization, and even death. So differences are there from year to year, partially because seasons differ and new strains may be more virulent, but also because vaccines that are developed every year may be less successful in triggering a lasting immune response to protect us effectively, which in turn undermines herd immunity. The world, meanwhile, is anxiously waiting for a vaccine for COVID-19. Many companies and academic institutes are involved in this race. Multiple different strategies are pursued. And this Nature review article here provides a really nice overview of that. Classical, live attenuated, and inactivated virus strategies have now made way to subunit strategies where viral components are leveraged to facilitate a lasting immune response. And new on the scene are also DNA and RNA strategies, where the human body is essentially treated with gene therapy to provide the viral components themselves to trigger the required immune response. Really clever science. In addition to these novel vaccine platforms, we also see innovation on, for instance, temperature stability and orally administrative technologies. Not trivial at all, as we want to inoculate 7 billion people across the globe and in their climate zones. The good news is that the collaboration is becoming the norm in this race to treat and prevent COVID-19. From hundreds of poorly designed, small, non-controlled clinical trials, we're now moving to joint large registry trials with much greater power. Regulators too are providing explicit support to encourage therapeutic and vaccine solutions through breakthrough designations and accelerated review times. Regulators are proving yet again that they are key partners in getting critical treatments to patients faster. We also see pre-competitive information sharing. And here I want to do a little shout out to the COVID-19 Pharmacology Research Center website, where we ourselves, but also the Gates Foundation and many other companies involved in this space are sharing their science as we speak. During the development of vaccines, multiple questions need to be answered. Foremost, there's a focus on safety. As we're going to expose billions of people to these vaccines, we better make sure that it's safe always. We also note that in order to accelerate development, dosing strengths and regimens are not always tested very widely during the process of developing vaccines. By the time that you gear up for phase 3, you really want to know what your large-scale manufacturing should look like. And that in turn results in the dose and the regimen getting locked in very early on into the process. Thus, after phase 1, where we look at safety and tolerability, and phase 2, where we look at safety and tolerability and initial immunological response, we typically enter phase 3, where we look at prophylactic efficacy with a single dose strength and regimen only. At that point, questions remain. Do we have the right dose and right regimen? Is it optimized for a lasting immune response? Do we need to revaccinate? And so, how often? Do different subpopulations have different dosing needs? All critical questions. And just like the recent ongoing race to repurpose existing drugs to actively treat COVID-19, we now too see that the dose selection in vaccine development is taking a lower priority. And that is a risky strategy in our minds. Accelerating vaccines for COVID-19 should not be about creating a safe vaccine as soon as possible, but rather a safe vaccine and an efficacious vaccine as soon as possible. Getting it right first time instead of the second, third, or fourth shot on goal. Production costs and the medical urgency simply don't allow for multiple shots on goal here. Over the years, modeling and simulation has had an increasingly impactful role in drug development. Before the year 2000, approximately, modeling and simulation was very much within the academic domain. But then in the early 2000s, we saw it make its entry in the form of pharmacometrics and systems biology, where we started to see larger and larger impact on key decisions in drug development in the pharma companies. In 2005, the seminal paper by Peter Milligan and colleagues came out, where we basically industrialized this approach and nicknamed it model-based drug development, which later on got further popularized by calling it model-informed drug development. And around 2010 to 2015, there were a series of EMA workshops where we also highlighted the important role of discovery and acknowledging that discipline too benefits from quantitative approaches. And at that point, we started calling it model-informed drug discovery and development, or MID3 for short. MID3 now is well embedded in today's modern drug discovery and development processes, but also is very much part of regulatory approval processes and the way the regulators look at the filing packages. It's also fair to say that vaccines are probably the last drug development area to join this MID3 revolution. So in today's webinar, we will showcase a few examples and potential solutions to provide a better understanding of expected prophylactic efficacy and immune response dynamics of vaccine development. As we are at the start of COVID-19 vaccine development, most of these approaches are untested for SARS-CoV-2, but have been established in other vaccines programs. So our hypothesis is that vaccine development too needs model-informed drug development, and to get it right first time. It will help to select the best vaccine candidates to prepare for the all-important manufacturing scale-up while we conduct phase 3. Application of quantitative modeling approaches with focus on model-based meta-analysis and quantitative systems pharmacology have proven their value, and they will be essential for COVID-19 vaccines too. With that, I would like to pass it over to Amy, who will now take us through the next section and explain some of the considerations that go into vaccine development.
Thank you, Thomas, for the introduction. So in the painting here, you can see Dr. Edward Jenner, who administered the first vaccination as a preventive treatment for smallpox, a deadly disease that had killed many people over the centuries. Since then, Jenner's model was employed by many scientists through the 19th and 20th century to develop new vaccines, such as whooping cough, hepatitis B, and many others shown in the blue box. The method for development and understanding the vaccine mechanism of action has improved over time. Commonly, vaccine is dosed to animal to ensure safety prior to human trial. In the non-clinical experiments, a starting dose is tested and escalated until the dose is no longer safe or saturated. This then provides a safe dose range to be scaled up and applied in the clinical settings. In human, the dose decisions are made based on escalation results and pairwise comparison of best doses. In addition, if a next-generation vaccine is available, a higher sample size might be required to allow comparison of vaccines. The time required for the stepwise approach from vaccine candidate to approval are often long. Much information and data are then left behind once the vaccine's development have moved forward or terminated. This results in opportunity where a model-informed approach can enhance vaccines development. With this framework, key questions are set at the beginning, and these questions are continuously refreshed and answered as data emerges. 3 levels of question can be asked to understand the disease, the vaccine, and the mode of action. And due to the novelty of SARS-CoV-2 and vaccines, assumptions and data gap must be identified to quantify the uncertainty. This assumption must be tested and validated as information emerges. MID3 also offers a range of modeling approaches such as MBMA and QSP to accelerate the vaccine development. As mentioned earlier on, the key question of COVID-19 vaccine development centers on the question around immunity and protection, dose and regimens, and competitor landscape and target populations. For example, what level of immunogenicity is required to achieve protection, and what is the corresponding dose and regimen? How many booster vaccines are required and how often to ensure lasting protection? And in addition to immunogenicity markers such as IgG titer, what other markers are related to response and efficacy? And what are the relevant target population differences, for example, in pediatric, elderly, and vulnerable? And in terms of COVID-19, which target to the whole global population, it is very important to understand whether maturation or deterioration due to age has impact to immunogenicity and protections, and also can we utilize and select from the high volume of publications every day from the literature to support this. With this, here we present a MID3 framework that can apply to COVID-19 vaccine and other vaccine development. In step 1 and step 2, a wide range of doses and formulation are tested in non-clinical species, for example, in rodent and non-human primate models, to understand immunogenicity over time. Through mathematical modeling, the developed model allows prediction and understanding of dose-response relationship. Historical vaccines in similar classes can also provide information, for example, for the dose range, and the prediction can also provide recommendation of how best to carry out the next non-clinical experiments to collect missing information to refine prediction and establish confidence. In step 3, the developed animal model is translated to human to predict human dose-response relationship using human immune response model. And in step 4, doses selected using the model is tested in the first time in man trial. As the clinical data emerges, this is fed back to refine the model. This learning and confirming through modeling inform approach continue as the vaccine move along the development phase. The established model can also predict the dose and regimen required to achieve protections. In step 5, public non-clinical and clinical information of other vaccines in different populations, including animals, pediatric, elderly, and other vulnerables, are selected and used to build a database. MBMA uses this data with the data of the investigational vaccines to predict relationship of immune response and outcome, to inform dose selection and various populations and allowed early head-to-head comparison. And in step 6, this allows acceleration for go-no-go and approval of vaccines in populations. One of the most important questions on vaccines development is the level of immunity required to establish protection and the duration of protection. This is a function of dose and when booster vaccines doses are administered. In order to answer these questions, the understanding of immunological memory is essential. On the left, a high-level graphic guide published in the Nature illustrates how body's adaptive immune system can learn to recognize new invaded pathogens such as SARS-CoV-2. This enabled us to develop a mechanistic mathematical model on the right to describe how a vaccine primed the adaptive immune system, including the generation of antigen-specific memory B cells and the duration of immunological memories. A COVID-19 clinical outcome database funded by the Bill and Melinda Gates Foundation has been developed as Atara. This is available through our web-based graphical interface Codex. The database is curated using established methodology with emerging trials and literature-reported data on all treatment effects, including anti-malarial, antiviral, anti-interleukin-6, etc. It collects patient characteristics and a variety of endpoints, including viral load, biomarkers, vital and clinical outcome that allows us to understand disease progression in patients and the treatment effects comparison. New treatment can be benchmarked against those that are currently being used in the clinic. The database is continuously updated as treatments emerges and will include vaccines information in the coming time. A dedicated webinar on this database effort will follow. The simplest meta-analysis is to generate a pairwise comparison between placebo and vaccines across multiple data sources. This does not enable us to compare different vaccines. To do this, we have to use a network-based meta-analysis where we can do an indirect comparison of vaccines by differences with placebos. However, this still treats different dose levels of the same vaccines as a different treatment. And so, to infer dose-response relationship, we have to use a model-based meta-analysis, not only allowing us to take dose level into account, but also covariates such as age, race, and comorbidities. Together with the database and MBMA analysis, we can inform decision-making in vaccine development. This ranges from dose and population selection, go-no-go criteria, to market positioning using virtual head-to-head comparison. In brief, MBMA incorporates parametric models based on literature data and in-house data. It quantifies the effect of vaccine, time, and patient population characteristics on the outcomes. It can include trial-level covariate relationships on the dose-response models to account for differences between patients' population. It also allows simultaneous modeling of multiple endpoints and can therefore link biomarker, for example, immunogenicity, to clinical endpoints, for example, in incidence rate or hospitalization. Like network meta-analysis, it can provide indirect comparison and simulations of head-to-head trials, but uses longitudinal dose-response model for individual vaccines or vaccines classes. It can also be used for simulations of trial and trial success predictions. Vaccines have been developed to protect people from viral, bacterial, parasitic infections. From an MID3 point of view, the analysis approach are very similar. We can make use of the wide range of data reported in the literature to understand the adaptive immune system. We will now illustrate the model-informed approach, including MBMA you just described, using 4 case examples of different antiviral vaccines and demonstrate how it can be applied to COVID-19. I will now pass the presentation to Jos Lomas to share this example.
Thank you, Amy. So as mentioned, we will now go through 4 examples, each of them addressing an important question for vaccine development. These examples demonstrate that the impact of MID3 can have at various phases in the drug development process, such as using non-clinical data, designing an optimal dose regimen, predicting the efficacy of a new vaccine, and extrapolating to long-term protection against viral infection. Examples 1 and 3 were also presented by Jeff Sachs at the PACE Pharmacokinetics meeting last year. So the first example is about making better use of non-clinical data to support translation to human. Non-human primate longitudinal antibody titer data at various doses was used to develop a model mimicking the adaptive immune system. The model described dose and time dependency of the antibody titer. In figure 1, the blue circles are the observations, the black line is the model fit, and the red line represents the geometric mean of the data. You can see the effect of a boosting dose, causing the antibody titers to rise to much higher levels, which is well described by the model. The response is saturated between the 2 dose levels that are shown. By calibration of the amplitude, the model was translated to human to allow for prediction of the immune response at different dose levels, regimens, and formulations. This human model was informed with phase 1 data and then used to influence phase 2 trial design by incorporation of a lower dose level and an additional regimen. So the impact of the work included an increased confidence in the ability to translate findings from non-clinical species, and also the ability to model the immune response of this virus K vaccine. It also changed the planned phase 2 design. The second example shows how MID3 supports selection of a dose regimen needed for a required immunogenicity response for a hepatitis B virus vaccine. In this example, data from 21 trials with almost 2,000 adult patients across 8 different hepatitis B vaccines was analyzed, reflecting over 10,000 post-vaccination antibody titer samples, a big data set. A fairly simple 3-compartment model was developed to describe the relationship between vaccine administration, memory cell growth, and antibody excretion. The model had 6 unknown parameters, of which 5 had to be estimated. The key assumption made was that the immune memory is assumed to reach a maximum after priming. 1 or 2 booster vaccine administrations may be needed to obtain full potential of the immune memory in terms of anti-hepatitis B antibodies. Simulations of scenarios allowed varying of dose regimens. The plots show a simulation of a typical study applying 3 20 microgram vaccine doses at months 0, 1, and 6. The precise rate of the antigen decay may be relatively unimportant in quantifying the rate of memory cell generation, as long as most of the circulating vaccine antigen is removed prior to the subsequent vaccine dose. The generation of memory cells is found to plateau after approximately 6 months in this example. The left-hand panel shows the estimates of 2 of the parameters related to the rate of memory cell generation for the various vaccines included in the analysis dataset, as well as the predicted time needed to obtain protective immunity. The average time to develop memory cells above 10 micro-international units per mil is the best indicator for the time taken to develop protective immunity after vaccination. plots at the right-hand side show model fits to the data. Each curve represents a different study. The 21 different studies with 8 different vaccines and different dose regimens were well described by this relatively simple model. So based on the modeling, it was concluded that a single vaccine dose could be sufficient to generate protective immunity. This could mean that additional immunogenicity trials could be omitted for the development and analysis of new vaccines to treat this disease. Next slide, please. In our third example, we predict the efficacy of a new vaccine X. First, literature data from clinical resources were collected. These data, together with the in-house data, were used as input to develop an MBMA model, which included both vaccine and placebo data. Optionally, we could have included non-clinical data to support model development. The MBMA model described the relationships between immunogenicity biomarker and clinical outcome based on immunological understanding. In this case, the clinical outcome was incidence rate of the occurrence of a viral infection. Covariates were included in the model to describe differences among populations, routes of administration, severity of the disease, and formulation options. In the next step, the resulting model was used to simulate a large population with and without vaccine. Then the efficacy was calculated based on the resulted predicted incidence rates for the two groups. If there's no overall difference between incidence rate of the placebo group and the incidence rate of the vaccinated group, then the efficacy would be 0, no efficacy at all. If all subjects in the vaccinated group would be fully protected, then the efficacy is 100%. Next slide, please. On the left-hand side, you see a grid of plots with biomarker values on the horizontal x-axis and reported incidence rates on the vertical y-axis. The various plots show the data for the populations A, B, and C, and different disease severity levels 1, 2, and 3. The size of the data points indicate the size of the trials. Now, in a single network MBMA model was built to describe all different vaccines and accounting for all populations and disease severity levels. In that way, all data points contribute and strengthen the biomarker incidence rate relationships for each population and disease severity level. On the right, the MBMA model shows the relationship for population B at disease severity level 2, with all literature reported data points scaled to this situation. The model shows that as the biomarker value increases, the incidence rate decreases. The yellow shaded area is the confidence interval of the model. Then this population B was simulated, obtaining a range of biomarker values when treated with vaccine X or with placebo. Based on the calculated incidence rates, the efficacy for a study with vaccine X was predicted to support a go, no-go decision for this particular program. So the MBMA model has provided a very powerful overview of all vaccines that have been reported in literature against virus X that has been studied so far, and also allows the new vaccine to position them along them. Next slide, please. The last example shows an analysis where immunogenicity data were extrapolated to assess long-term protection by a vaccine against Japanese encephalitis. First, a model was built to describe the antibodies change over time. The model was subsequently used to predict the duration and extent of serological protection. A piecewise linear mixed model was chosen because it provided the best fit of the model to the data and also to the antibody decline from the observed peak. Individual seroprotection was based on the accepted threshold that the antibody titer should remain above 10. Immunogenicity data from a randomized clinical trials using 1 or 2 dose levels of the vaccine in healthy volunteers was measured at various time points up to 5 years. The box plots show the observed antibody titer between this time 0 and 5. In subjects receiving a single dose of the vaccine, The green line represents the accepted threshold of the protection of 10 times dilution. The plot on the right-hand side summarizes the protection against the virus we can expect based on the titer observations and threshold. Next slide, please. The spaghetti plot on the left-hand side now shows the population prediction, that is the black line, and individual fits, the blue lines, of the antibody titer of 99 subjects using this piecewise linear model. The population average closely matched the observed median antibody titers observed up to 5 years. The red segment in the plot indicates the observation time, and the blue segment the extrapolated time. The plot on the right-hand side now shows the prediction of the proportion of subjects that will be seroprotected through time with 95% confidence intervals. The open circles represent the observed seroprotection values. Using the assumption that the log linear rate of the antibody continues after 5 years, the majority of the subjects will be protected for at least 10 years after a single dose vaccination. These were the 3 MID3 examples I wanted to share with you. Now we will continue with Piet van der Graaf's part showing the capabilities of quantitative systems pharmacology in vaccine development.
Thank you, Jos. In the final part of this webinar, we will focus on the application of quantitative systems pharmacology for vaccine development. Quantitative systems pharmacology, or QSP, can be considered as an extension of the MID3 methods that were presented earlier in this webinar. QSP combines the well-established pharmacokinetic, pharmacodynamic principles and methods from pharmacometrics with detailed mechanistic models of targets, pathways, and disease from systems biology. A QSP model can initially be used to simulate outcomes before clinical data, building the model with literature data on the biological system and in vitro and preclinical experimental data. Once clinical data becomes available, the QSP model can be updated in a continuous learn, predict, and confirm cycle. Pharma has recently started to use QSP as illustrated by this white paper from the US IQ Consortium, which presents some 45 case studies of the application and impact of QSP in drug discovery and development. QSP has also started to play a role in regulatory science, as illustrated by the graph on the right, showing a year-on-year increase in the number of FDA submissions with a distinct QSP element. However, QSP has not been widely used yet in vaccine development, and in the next part of the webinar, we will present the recent progress we have been making in this area. The reason we became interested in this is that we and others believe that QSP can play a new and important role in improving the vaccine development paradigm. Specifically, QSP can be used to simulate vaccine response in virtual trials before any clinical testing. It can help to optimize the vaccine itself, the dosing regimen, and the adjuvant. In early clinical testing, QSP can scale responses from healthy volunteers to a wider population, and subsequently it can be used to optimize treatment in special populations and for pharmacogenomic-driven personalized dosing. Our journey in this area started some 4 years ago, when together with 7 major pharmaceutical companies, we established the first of its kind QSP consortium to develop a highly mechanistic QSP platform to model the immune system, and in particular its response to foreign proteins. The consortium has just successfully completed its first 3-year cycle and has now entered its second phase to expand and validate the model further. The focus of the consortium has been on the utilization of this industry-leading QSP platform to predict and manage the undesired immune response to biological therapeutics, which is a major issue in the clinical development and therapeutic use of, for example, monoclonal antibodies. As I'll show you later, more recently we have started to consider the use of this QSP platform in the development of vaccines. This slide shows a high-level overview of the model and how it has been integrated in a bespoke QSP software platform. In the top left corner, you see an actual screenshot of part of the model in the format of a so-called biological map, which is a detailed graphical representation of all the biological elements and processes included in the model. The various cell types and other model species are factored in a three-compartment physiological model comprising lymph, vascular space, and plasma, as summarized in the table on the right. The QSP model is fully integrated with the SimCip PBPK simulator platform, which allows for integration with physiologically based pharmacokinetic modeling, as well as the generation of virtual patients and virtual trials using the extensive special population databases that have been developed over many years. A wide variety of different inputs can be fed into the model, such as informatics data, in vitro experiments, as well as population and actual clinical data. In the interest of time, I will only demonstrate how information on the structure of a protein or vaccine can be used as a starting point in the next few case studies. Let's take the widely used monoclonal antibody Adalimumab as an example. On the left, you see a comparison of part of the structure of Adalimumab against the sequence of the full human genome, highlighting two areas in red, which would be recognized by the immune system as foreign or non-self. These potential immunogenic sequences can then be analyzed using other dedicated bioinformatics tools to predict binding affinities against epitopes of the human leukocyte antigens, HLAs, such as DRB10101, shown in the top row of the table on the right. The frequencies of the HLAs can be varied based on, for example, population data, or even actual individual HLA genotypes. Based on this input, we can now run virtual trials with our QSP platform and compare the predictions with actual clinical results. In the graph, the thick lines are the QSP model simulations, and the thin lines and open triangles the results from the clinical study reported by Bartels and co-workers. On the left, you see that the model accurately predicts the impact of the formation of anti-drug antibody, or ADA, on the time course of the pharmacokinetics of Adalimumab. We have now validated the model in a similar way against clinical data for approximately 20 biotherapeutics. For the purpose of this webinar, however, the graph on the right is the most relevant one. This shows the development of ADAs over time following Adalimumab administration. Again, the model predicts the time course and the magnitude of the ADA response well, which gave us confidence that the same approach can be applied to vaccines, where the terminology now changes from ADA to neutralizing antibodies in response to vaccine administration. As a first proof of principle validation of this concept, we focused on the Hepatitis B HPV vaccine. Using the same approach I showed you earlier for Adalimumab, we analyzed the sequence of the HPV protein, which was used to develop the actual vaccine, to generate the HLA epitope binding table shown on the right, which was then used as input for the QSP model, as I showed you before. One difference with the first case study that needs to be considered for vaccines is the effect of adjuvant. Our QSP model captures this in a module where adjuvant acts as a maturation signal to activate dendritic cells, which can be calibrated with preclinical and clinical experimental data. With this input, our aim was to see how well the model could predict the actual clinical trial results using the same dosing regimen of 3 subsequent vaccine administrations. The results are summarized in this slide, which shows the model predicted percentage of patients showing a clinical response to the vaccine on the left, compared with the actual clinical results on the right. I should point out that we did not try to calibrate and optimize the model any further at this stage, but we concluded that the results look very encouraging and provide a first proof of principle validation of our QSP approach. In the last case study, we will illustrate briefly how the QSP platform can be used in the design of novel vaccines ahead of clinical testing, or even before preclinical data has been generated. It will probably not surprise you that we picked COVID-19 as an example. The structure of the virus was published soon after the outbreak, and we generated a hypothetical vaccine based on the receptor binding domain of the spike protein, highlighted in red, following the same approach as shown in the 2 previous case studies. We did not include any adjuvant component in the first instance, but of course that could be done in the same way as was shown for the HBV vaccine if data was available. The simulation on the left shows the actual predicted seroconversion following a single administration of our hypothetical vaccine, and the right panel the average time course of the neutralizing antibodies. To illustrate how such a model could be used in early clinical development, we compared QSP model predictions between a healthy volunteer and an elderly population, which shows that in the latter population, the efficacy is approximately 20% lower. Other applications could include the use of patient-specific HLA genotype data for personalized dosing, as well as the identification of biomarkers to guide dosing and clinical development. In summary, I hope the 3 case studies I presented have shown how quantitative systems pharmacology can be used in vaccine development. QSP can be applied before and during clinical testing to optimize the actual vaccine, adjuvant, and dosing regimen. It can also be used to extrapolate between different populations, predict the impact of disease and medication, and ultimately provide a route towards rational personalized vaccine treatments. At this point, I would like to hand it back to Thomas.
Thank you, Piet. Really interesting how you want to apply that in the COVID-19 space there. So today, let's wrap up and draw some conclusions on what we presented here in the last hour. So we aim to convince you that yes, vaccines absolutely need to be safe foremost, but we shouldn't forget that we also need efficacious vaccines. The urgency and the medical need is too high to not answer key questions such as, do we have the right dose regimen to get the optimal level and duration of prophylactic protection? And if not, do we need to revaccinate or use initial booster regimens? And do all subpopulations, such as elderly, people with underlying diseases, aka comorbidities, require the same regimen? We hope to demonstrate that vaccines too will benefit tremendously from model-informed drug discovery and development techniques to answer these key questions. Specifically, model-based meta-analysis is a powerful tool to translate immunological response to prophylactic protection and conduct comparative effectiveness assessments in search for the best vaccines for subsequent global scale-up and global rollout. Quantitative systems pharmacology approaches established initially for unwanted immunogenicity of biologics can now be repurposed to predict wanted immunogenicity responses of novel vaccines and describe the mechanisms of novel vaccine platforms. By bringing these state-of-the-art solutions to the table now, we can jointly assure that we can tackle COVID-19 and get it right first time. I think with that, I would like to progress to the next and last slide. It's just a small part of the team on Certara side and that helps us provide the materials for this webinar. Also, of course, would like to acknowledge our many clients that we collaborate with and where good cross-pollination of science is occurring every day. I think it's honest to say that we're actually excited to be able to fight in this unprecedented crisis. I mean, this is the reason why we became scientists in the first place. And we do want to further emphasize that we can't do it by ourselves. We need to do it jointly. So again, calling out here for further collaborations between the pharma companies, regulators, the NGOs, the academic teams. Please make use of the COVID-19 Pharmacology Research Center as well. And with that, I would like to go to the Q&A section. We'll try to answer as many questions as we can within the time frame. If there are any additional questions afterwards, please don't hesitate to reach out to me, and I will endeavor to connect you with the right experts as soon as possible. We're here to help you. With that, Suzanne, I think you will have the questions for us, correct?
That's right. That's right, Thomas. Thank you so much, Thomas, Pete, Amy, and Jos. Looks like we've got a bunch of questions from our audience. Our first question is for Dr. Chung. Does it imply that in the absence or insufficient level of immunoglobulins following vaccination, are memory cells adequately activated? If the memory cells were adequately activated, would you still achieve immunity following infection?
Yeah, thank you, Suzanne, and thank you for the questions. And I think for this, we need to really understand whether this level of immunoglobulin, for example, in IgG, can activate the memory B cells to achieve for immunity or say the protections. We need to do so by exploring the relationship of these levels, which can be resolved from a particular vaccines, and other markers that were collected, which can surrogate for the protections, for example, like interferon gamma, T cells, and also the same with the outcome after the vaccination when the data emerges as well. And non-clinical data can also provide some ranges of immunoglobulin and with these immunomarkers, and we can assess how this relationship can translate to human. And using the modeling, MBMA, we can combine any new data from non-clinical and clinical data available from different vaccines of different classes, and also whether immunity can achieve at the same time. The duration of how long is this immunity is also very important, whether a booster dose is required or whether one dose would do it all. So with the model developed, that allows us to predict how long would the protection and what doses would generate the required immunoglobulin that it can achieve the protections. Thank you, Suzanne.
Thanks, Amy. I've got a question for Jos. How do the hepatitis B model predictions, hepatitis B vaccine virus model predictions compare with the clinical data?
Thank you for the question. So I tried to show actually, but maybe it was not clear enough that the actual data, they were kind of included in the hepatitis B example. And yeah, there was a good fit, so to say, of the model prediction with the data. So yeah, I do not have so much to add to that actually, sorry.
That's great. Got another question for you. Is the translation of the animal model to humans based on including covariates in humans in the course of model development using non-human data? If so, what would be examples of such covariates in this case?
Okay, thank you. Yeah, so I indicated at least you need to make distinction between animals and humans. They're not the same. So the most important adjustment that we made actually for this translation was adjusting actually the amplitude. Actually, in this particular case, we had to change actually the level actually that the so-called incidence was different in animals in comparison to humans. But also other aspects we need to take into account. So also the biomarker value may shift actually to different values for the animals, and that can be kind of strain specific. So yeah, it depends actually. After an exploratory analysis, you can probably deduce actually what kind of covariate effects you need to kind of include in the model.
Thanks, Jos. Thomas, someone wants to know what type of vaccine shows the highest success rate?
Well, isn't that the multi-billion-dollar question? I don't think we really have a single answer for that. I think it's a really interesting question, and everybody is thinking about this, especially specifically now in the race for COVID-19. I would argue that if you look historically, different diseases have different success rates with the vaccine platforms, but most importantly, some of the novel platforms we don't have that much information on. So live attenuated vaccines have been around for a long time, but on the other end of the spectrum, when we look at DNA and RNA vaccine platforms, it is really brand new. And that is important because there's also the manufacturing and scale up in there, and the knowledge around the more classical approaches may be beneficial there in the success rate. That said, I think from a purely theoretical perspective, the science behind a DNA and an RNA vaccine should allow you, in theory, to get a more sustained immunological trigger, and that would allow you to not have to do booster regimens, not have to worry about a longer lasting immune response, as long as indeed that DNA component remains built into the human DNA, and the virus component is indeed produced by ourselves to trigger the immune response. So I do think there are definitely benefits to the novel strategies. The proven strategies are useful. I think the good news is the fact that there are 115 different candidates in development. We are doing things in parallel within the industry to make sure, together with the academic institutes and hospitals, to make sure that we try different strategies here. And we may end up with multiple different vaccines tackling this.
Thanks, Thomas. Pete, someone would like to know, they state that the QSP model only shows the concentration of antibodies in the plasma. They wanted to know whether we have a disease model for COVID-19, because they state if there isn't one, it's hard to assess the vaccine just based on seroconversion.
Yeah, so first, that's a very good question. So I only showed the plasma concentrations, but we can simulate concentrations of neutralizing antibodies in all the compartments in our model. But for the sake, in the interest of time, I only showed plasma. The next step would be indeed to link such a model to an actual disease model, like COVID infection. So we haven't done that yet, but it's perfectly feasible, particularly given the modular nature of QSP models, where we can really kind of use different model elements as building blocks and kind of link them together. Thanks.
Thank you, Pete. There's another question for you. Someone would like to know where the Immunogenicity QSP platform can be assessed from.
You could contact us, and then we're more than happy to talk about it. It is not publicly available. We are certainly happy to talk to interested parties how we could use our platform in your programs.
Thomas, someone would like to know whether you are able to determine what physiological factors are responsible for the difference in exposure and response seen between healthy and special populations.
Yeah, so I think it's a good question, and it touches upon many of the quantitative solutions that we demonstrated here today, both on translating from preclinical to clinical, then from early clinical to clinical outcome, but also it's a QSP question in the very nature. I think a lot of the differences between the populations are driven from the fact that the immune response innate is different between those populations. So take, for instance, the elderly, the frail, their ability to fight off a disease is less typically because their immune system is just simply not as good anymore as it used to be compared to healthy young volunteers. And as a result, in vaccination, where you're trying to trigger this immune response, the response may also be less or less long lasting. And so I think those are typical things that we would explore within a QSP setting as well, trying to figure out, well, if we don't know yet, if we don't have clinical data, what if the immune response would be less because there's a quicker cell depletion, there may be a slower response rate between the cascade of events, what would it look like? And does that make sense in comparing to then the clinical data that emerges. And maybe, and Pete, I don't want to put you on the spot, but I do think you may have a few comments on that as well.
No, absolutely. Thanks, Thomas. So I fully agree that I think QSP can really help with the question regarding special populations. And I showed one, proof of principle case study where I showed the hypothetical response in healthy young volunteers versus elderly, and even making some simple changes to the model, you can see that the response is already quite different. It's multifactorial. It starts off with the PK of the actual vaccine, of course. HLA frequencies, as I pointed out, is different, not even across ages, but across different countries. The actual disease state, which will influence immune system, as well as co-medication. So I think it's a real multifactorial problem, and there's no single answer to it.
Okay, thanks, Pete. Got another question for you, Thomas. Can you specify differences in studying safety for therapies versus vaccines?
Well, I guess that's a broad question, a good question. In general, if we look at the history of vaccines, they're actually well established to be safe, and that means that if you want to demonstrate that indeed you're not observing any unexpected rare safety concerns, tolerability concerns, you do need to study these things in very large trials. And that's exactly what's happening in phase 3, where 30,000 patients is not uncommon to include in a phase 3 trial. It also has to do with the length of the trial itself, where people may get their vaccination, and then later on, if there's any safety concerns that develop further down the road, you need to follow them for quite a while. So there are involved studies. And I think if we look at the therapies, that's very much a mixture of many different molecular modalities, and they all carry their own safety risk-benefit profiles. Especially with the repurposed drugs, we already knew quite a lot about them, and that was helpful. So we already knew about the safety profiles of the repurposed drugs that are being used in the active treatment of COVID. That said, we hadn't applied them in this COVID setting, and these patients that are very sick and also going through a disease progress that starts out with a virological phase, then followed by an immunological overreaction phase, that too is different. And so I think when you evaluate active therapies in the setting of COVID, you really need to have a good understanding of the disease progression, the state that individual patient is in before you start treating them. And so I don't think there's really a good comparison to be made between what safety assessments for vaccines and active treatments do. You do need to make sure that the setting in which you're doing it, either with the active disease going through different phases, or in the prophylactic setting in a large population that is essentially a healthy population at that point, does warrant good safety controls in there. And again, that's why the trials are lengthy. That's why the trials are large, which is not, it's a balance there. I mean, the world is waiting for these vaccines, and at the same time, we need to make sure that they're safe.
I agree completely, Thomas. Thank you. Jos, we've got a question for you. Someone wants to know whether the model you discussed works for all routes of administration or just injection.
Yeah, so in principle, the model works for all routes of administration, but there can be differences depending on what kind of route is taken. If you think about IV versus IM, there can be a kind of delay actually. And also, there's the time course actually that certain biomarkers will rise actually and give protection. It can be different. So therefore, that's always an element actually in our modeling, if relevant, that we take into account if we should account for differences in the route of administration that's kind of applied. So the answer is, in short, yes, we do account for that, definitely.
Great, thank you, Jos. Pete, someone wants to know whether there's any pharmacology that can't currently be included in the QSP models that could be important.
I'm tempted to say no, I can't think of any, but that may sound a bit too overly bullish. But in principle, I think, as I mentioned before, we can include any mechanism as kind of modules to models like this. It obviously depends to a large extent how well we understand the pharmacology or the mechanisms that we would like to add. But if we have a good understanding of a particular mechanism, and ideally we have some data that we can use to build the model, in principle, I don't think we draw the line somewhere. Certainly, years ago, some people argue that immunology was probably one of those examples which you couldn't model because it was too complicated. But now I think, well, hopefully I've shown, we've made great progress there. We're doing a lot of work in neuroscience now, for example. Again, years ago, people said the brain is just so complex you can't model it. And I don't think we can model the whole human brain, but we can certainly model kind of elements of it fairly well now.
Thank you, Pete. Looks like we have one final question for Thomas. Regarding DNA and RNA-based vaccines, should we be concerned that these vaccines might have carcinogenic potential?
Yeah, I think that's an excellent question. I mean, with any novel drugs, any novel modalities, that's a question that's being asked in drug development anyway. And I think in a situation where we do essentially gene therapy and building in DNA structures within our own DNA, that's definitely is a question that needs to be investigated, and it's part of the regulations in how these things are developed. So I think it's an important checkpoint, but I would also argue it's an important checkpoint with any other drug development.
Wonderful. Thank you so much to our speakers, Thomas, Amy, Pete, and Jos, and thank you so much to the audience for all those excellent questions. Next slide, please. We have a few short announcements before concluding the webinar. On July 14th, we will be presenting Exploring the Effect of COVID-19 on HTA Bodies Now and in the Future. Finally, on July 16th, Dr. Jaap Mandema from Certara will present Model-Based Meta-Analysis for Comparative Efficacy and Safety of Therapeutics for COVID-19. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.